Good day, I'm Cesar, and my topic for today is about eukaryotic cells, prokaryotic cells, and their difference. But before that, I will just tell you a short story. I'll never forget a circular red spot I developed on my arm when I was in elementary school. It left a lasting memory in my mind because it was something called ringworm, and with my active imagination, I thought I was now infected a ring-shaped worm. I learned you've got to be careful about names because ringworm isn't caused by a worm at all. It's, it's actually a fungus which is turned out is pretty common and can be carried by many things like pets or soil. And since up to that point, I was used to antibiotics as a way to treat infections. I assumed I'd be given antibiotics, but I wasn't. I was given an antifungal cream instead, and it went away. Yes, it's gone. So it made me wonder, what made it different from the bacteria that had me sick in the past? Why wasn't I given antibiotics? Well, antibiotics target bacteria. Antibiotics can destroy bacteria by affecting their ability to reproduce, damaging their cell walls, or interfering with their ability to make proteins that they need to survive. Just some examples. But it turned out bacterial cells and fungal cells are very different cell types. In fact, fungal cells have more in common with your cell, which are animal cells, than they have in common with bacterial cells. And that has a lot to do with the comparison of prokaryotic cells with eukaryotic cells, which is what we will focus on. First, just a refresher, recall that the modern cell theory includes the statement that all living things are made of one or more cells. All living things. In the three domains of life, prokaryotes are organisms that can be bacteria and archaea. They are unicellular, which means they are single-celled organisms. Eukaryotes are organisms that all fit in this last domain, eukarya. Eukaryotes may be protists, plants, animals, or fungi. They can be unicellular or they can be multicellular, which means they can be made up of many cells, like you. By the way, just to clarify, the word prokaryote is typically used to refer to the organism itself. When you are describing its cells, you are describing a prokaryotic cell. Same for eukaryote. Eukaryote typically refer to the organism itself, and when you describe its cells, those are eukaryotic cells. Prokaryotic cells and eukaryotic cells do have a lot in common. Both have DNA. That's critical because DNA is the cell's genetic material. But prokaryotic cells and eukaryotic cells have ribosomes, which are small organelles, and organelles being like a tiny organ. Ribosomes have the important job for making protein. Got to make protein. Both cell types have cytoplasm, the jelly-like fluid within cells. Both of them have the cell membrane, also known as plasma membrane, which is critical because it controls what goes in and out of the cell and therefore maintaining homeostasis. All cells have a cell membrane. Now, as for cell walls, most prokaryotic cells have cell walls. Many eukaryotic cells, plant cells, and fungus cells, for example, can have cell walls. But there are plenty of eukaryotic cells that don't have cell walls, such as animal cells. But what makes prokaryotic cells and eukaryotic cells different is especially interesting. Eukaryotic cells are more complex than prokaryotic cells. They tend to be larger than most prokaryotic cells. And to help me remember some more difference in this next part, I like to remember that pro in prokaryotic or prokaryote rhymes with no, and you in eukaryotes rhyme with do. Prokaryotic cells have no nucleus to contain their DNA, so you will find their DNA is not contained within a nucleus. It's a bit messy here. They have no membrane-bound organelles. Membrane-bound organelles are fancy organelles that have their own membrane like the nucleus. Mitochondria, the endoplasmic reticulum, and the Golgi apparatus. A big indicator of eukaryotic cells in this nucleus, eukaryotic cells do have a nucleus to contain in their DNA. Depending on what type of eukaryotic cells it could have different types of membrane organelles. For example, a plant cell is likely to have chloroplast while an animal cell would not. All cells of living things fall in one of these two categories. And understanding the characteristic of these two cell types can help us better understand the diversity of living things, whether they are archaea, bacteria, protists, fungi, plants, or animals. And in the case of my example or my story, realizing whether an infection you're dealing with involves prokaryotic cells such as bacteria or eukaryotic cells such as fungus. That's all. Thank you. Bye. Taxonomy. There are a lot of animals that I have always thought are cool. But one day, in my 7th grade science class, my teacher told us we would get to see one of the most amazing animals ever in class, a hydra. And I have to confess what I imagined in my head and what actually was reality. It was a little bit different, but I'm kind of used to that. Turns out hydra are pretty cool. Hydra are animals that are very small, a few millimeters in length. They live in fresh water. They can attack and eat their tiny swimming prey, and they can reproduce by budding an identical offspring on themselves. Excuse me. But if you saw them, you might not think they are an animal. What makes us categorize them as an animal and not a plant or a protist or a fungus? In fact, how do we classify in the first place? Well, this let us jump right into our next topic of classification. Taxonomy includes the naming and classification of a species. But back in time, the 18th century, there wasn't a separate category for other organisms like protists or bacteria. They didn't know about detailed cell structure differences. They didn't know about DNA, so they didn't know about genetic relationship that now can determine how we categorize organisms. And that's the thing about classification. As we learn more about DNA and therefore relatedness, how we classify organisms can continue to change. So first, let's look at an awesome hierarchy system, mnemonic. Their king, Paramecium, care only for green spirulina. But we sort of have a thing for protists and we realize that might not be as memorable. So let's now take a look at each group of this hierarchy, starting with one of the most exclusive group, domains. Domains. It's so awesome that all of life will fit into them. So there are 3 domains, bacteria, archaea, and eukarya. The domain bacteria is full of bacteria. There are prokaryotes and therefore have characteristics we've mentioned of prokaryotes before. This can include bacteria that make me sick, the bacteria that are in your intestine and my intestine, helping you digest, and me also, the bacteria helping with decomposing, the bacteria fixing nitrogen in the soil, tons of different kind of bacteria. Archaea and prokaryotes too, but they have some major DNA and structure differences that give them their own domain. And while they may seem more closely related to bacteria since they are prokaryotes, recent DNA evidence linked them to having more in common with eukaryote, which is interesting. Many of Archaea are extremophiles. That is, many of them like the extremes. Some like extreme salt environments, for example, which mean they can handle extremely salty environments like the Dead Sea, or methanogens. They can live where there is very little oxygen. In fact, most of them can't even handle oxygen. They use carbon dioxide to make their energy instead and produce waste gas, methane. Some of them live inside animals that eat a lot of cellulose, like cows or termites. Another extremophile is thermophiles. They like extreme temperatures. If you ever dream of living near the deep sea hydrothermal vents, well, if you were a thermophile, you'd be in luck. We mentioned that archaea and bacteria domains are separate because they have some major DNA and structure differences that are significant enough to separate them. And so does the third domain, eukarya. These are eukaryotes, and so they have characteristics we've mentioned before that are common for eukaryotes. And that's where we'll focus now. So the next level, less exclusive and more specific than domains, is the level of kingdom. Here's the big disclaimer about kingdoms. Its organization is often changing, and it's not even something that all scientists agree on. We've seen a 5-kingdom system that looks like this, and a 6-kingdom system that look like this. And to be honest, it's a changing view as we learn more DNA and cell structure evidence. But if we focus on these eukaryotes, let's touch of them briefly. Protista, extremely diverse, and there is often talk about dividing it because of how diverse it is. There are protists that are animal-like, and protists that are plant-like, and protists that are fungi-like, but many scientists don't consider them to quite meet the requirements to be in these kingdoms. Protista includes both autotroph protists, making their own food, and heterotroph protists, which consume other things for energy. Most protists are unicellular, but they can be multicellular. Some have cell wall made of cellulose, like plants. Some don't. Fungi are heterotrophs. If that's hard to remember, just think about athlete's foot. It's a fungus on your foot, in my foot, and it's not doing any photosynthesis there because that'd be really weird. Nope, it's there, causing irritation, eating these skin cells. Fungi are usually multicellular, but they can be unicellular. Must have cell walls. It's a carbohydrate. Plants are autotrophs. Yes, even the carnivorous plants. because they still make their glucose from sunlight energy. Plants are multicellular and they have the cell walls of cellulose. And finally, last up, the Animalia. Animals. Okay, Hydra, you can come back. This mostly multicellular and heterotrophic kingdom is the kingdom to which you belong. So now we have the other hierarchy level. We get less exclusive, therefore more specific, as we move down to the Hydra's phylum class, order, family, genus, and species. Because the species name is the most specific you can get, it is the least exclusive. The two-part naming system that we use uses Latin or Greek roots. This is its scientific name. See the first name? That's its genus. It's written with a capital letter at the start and it's written in italics. See the second name? That's the specific epithet, which is a fancy way of saying that it refers to one species in the genus. It has a lowercase letter and is also written in italics. So why do we care about the scientific name? Well, you could come up with a lot of common names for an organism that are very based on location. Take this mountain lion for example. It's also known as a puma, cougar, or Texas panther. Same animals, different names. But the scientific name here is a specific and recognized regardless of your location. And that's give power to an awesome way to organize and name species. That's all, thank you. Bacteria. When I was a little kid, my parents would let me take my favorite set of dinosaurs into the tub, probably to persuade me into taking a bath. I remember my dinosaurs well. In fact, I used to know of all of my dinosaurs. I had an epic dinosaur battles. Sometimes my sister will play with them too, a little differently. Anyway, I remember that one day, unfortunately, my favorite Stegosaurus lost his head. I mean, literally, poop off. And while I was determined to get my parents to fix it, we discovered that there was a stuff growing in the toy. Now I realize what was actually large enough to be visible was likely mold. But at the time, my mother told me that Surgeon Stegosaurus would have to retire from the bathtub and I had to get a new one because bacteria had taken over. It led to two misconceptions I developed about bacteria. One, that bacteria were only found on or in things that had gotten contaminated somehow, and two, that bacteria are always bad and that is why Surgeon Stegosaurus required replacing. Those are both not correct. First of all, these probably were bacteria in a dinosaur along with the mold. But that shouldn't be surprising because bacteria are everywhere. Bacteria are found in our houses. Yes, even very clean houses. And they are found outside. Bacteria colonize our skin and our digestive system. That addresses the first misconception that you only find bacteria on contaminated or dirty surfaces. And as for the second misconceptions, well, bacteria sure do get a bad reputation. Now, I'm not disagreeing that the toy should have been thrown out. That was a good decision though. There was probably mold that was in the toy dinosaur, a fungus, but there was likely plenty of bacteria growing there with it. A growing community of mold and bacteria. not ideal for a bat toy. But we don't want to mention that many times. All bacteria are lumped together as a bad things, which shouldn't happen, because not all bacteria are bad. Yes, in fact, many types of bacteria are helpful for organisms and ecosystems. We give you some examples. Bacteria that colonize your skin are beneficial and actually help keep harmful strain and other types of pathogens from growing. Bacteria in your digestive system actually help break down food, and some can produce certain vitamins. Some types of bacteria are used in producing some foods that we eat. In ecosystems, bacteria have a very important roles as decomposers. Bacteria are also major roles in the nitrogen cycle to fix nitrogen that plants need. These are just a few examples of helpful, beneficial bacteria. Now that's not to say bacteria can't be pathogens. Bacteria are the cause of strep throat, tetanus, tooth decay, some forms of pneumonia, diphtheria, salmonella, cholera, and many more. Antibiotics can be used to combat some of these. And while antibiotics are very important for destroying bacterial infections, we shouldn't leave out saying that some broad-spectrum antibiotics can harm the good bacteria as well. Also, we should mention that antibiotics do not work on viruses. Viruses are pathogens that are not made of cells at all. Additionally, there are vaccines which can prevent many types of both bacterial and viral infections. Okay, so what are the bacteria exactly? In the three domains of life, bacteria encompass one of them. They come in different types of shape, as you can see here. Some of these are heterotrophs, meaning they consume or feed on some organic matter. But some are autotrophs. They can make their own food. Plants aren't the only one that can be autotrophs. Let's take a look at a bacterium. Bacterium is just a singular word, while bacteria is plural. A bacterium is a prokaryotic cell, which are generally much smaller than eukaryotic cells, like ours. If you recall from our prokaryotic versus eukaryotic cells video, that means bacteria do not have a nucleus or other membrane-bound organelles. But you will find ribosomes, cytoplasms, a cell membrane, and nearly all bacteria have a cell wall. Like all living organisms, bacteria have DNA. Bacteria DNA, while still double-stranded, is arranged in a circular shape. Depending on the species, bacteria can also have a flagellum to help with movement, a capsule which can help give them extra protection or attachment abilities, or pili which can help with attaching to surfaces, including each other. Oh, and many bacterial species has a plasmid, which is basically like extra DNA. More about that later. Bacteria have some intriguing abilities that are different from our human cells that we'd like to mention. Unlike our own body cells, which perform mitosis and cytokinesis to divide, bacteria generally multiply even faster in a process called binary fission. This is a type of asexual reproduction when the bacteria can easily divide to make a copy of themselves. Since it is asexual reproduction, the daughter cells would be expected to be identical to the parent cells unless there is a mutation. Some types of bacteria do have the ability to share genetic material with each other. Remember how we mentioned that bacteria can be a plasmid, an extra copy of DNA with usually just a few genes on it? Bacteria can share these plasmids with each other in a process known as conjugation. The pili can be used to share this genetic information with each other. The pili can be used to share this genetic information with each other. If the plasmid happens to have a gene that gives some degree of resistance to antibiotic, this may allow the bacterium that received the plasmid to survive exposure to that antibiotic, which could be very problematic. You can learn more about how antibiotics resistance can develop over time in bacteria. Bacteria can also pick up plasmid from their environment. Often, when this happens, it is during a time of stress for the bacteria. In a process known as bacterial transformation, scientists can use a type of stimulus, such as a heat shock, to induce bacteria in a lab setting to pick up genetic material. There are all kinds of uses for this genetically engineered bacteria that you can explore. Some types of bacteria can form endospores. Endospores allow bacteria to be survivor in all kinds of hostile environment. Lack of nutrients, freezing temperatures, drought, just some examples. This is the reason why hospitals have to be a very good at sterilization process. We won't go through the process of endosporulation or how bacteria reactive after forming endospores, but this is definitely something interesting to explore. Finally, some type of bacteria, along with other types of prokaryotes called Archaea, can't be extremophiles. Unlike our own cells, extremophiles can live in extreme environments where there may be excessive heat, chemicals that our cells would find toxic, or even radiation. Overall, we share this planet with so many kinds of bacteria that scientists continue to learn more about them every day. And if this kind of topic interests you, you may want to look in the study of microbiology. So many careers, from agriculture to the medical field to environmental work, rest heavily on the study of microbiology. That's all, thank you. Archaea. Have you ever imagined things with a movie thriller voice? You know, the dramatic voice that commands you to listen and be intrigued that shows up on some movie thriller? We think it could actually work great for biology. An example, in a world with no oxygen, one little yeast cell will do fermentation. Okay, tell me, that does not make you want to learn at least a little about fermentation? If so, we have a video about that. And the yeast, which is a fungus, which can do it. But you know what would be the best topic for an exciting movie thriller? Archaea. Because they are probably some of the most exciting unicellular organisms out there. And many of them can live in an extreme environments too. By extreme, I mean think harsh but then more harsh. Areas with extreme temperature or high salt level. Though I should point out Archaea are not the only extremophiles. Bacteria can be extremophiles. It's interesting because when I first started teaching, it was common to call Archaea Archaea bacteria. But that is an outdated term because archaea aren't bacteria. Now, they do have something in common with bacteria. They are both unicellular. They're both prokaryotes, so they have no nucleus. And they also don't have membrane-bound organelles. They both have genetic material and cytoplasm and ribosomes. Both bacteria and archaea can be classified as autotrophs or heterotrophs, and they both do binary fusion to reproduce. Still, bacteria and archaea are two completely different domains in the three-domain system. That's a pretty big separation. What makes them different? In the three-domain system, you have the domain bacteria, the domain archaea, and then the domain eukarya, which has all the eukaryotes, plants, animals, fungi, and the protists. So let's look at the difference between bacteria and the archaea. Membrane structure is a very different. Remember that all cells have a membrane. It's really important for homeostasis. If you take a look at our further reading suggestion, you can also explore how the chemistry of the cell membrane of the domain archaea is different. One example is that archaea membranes typically contains either linkage instead of ester linkage. You would find ester linkage in the membrane of bacteria and eukarya. Some archaea have a lipid monolayer instead of bilayer. Turns out that the monolayer, as well as some of other structural differences of archaea membranes, can be useful for surviving in extreme environments. You will also find a unique cell wall in archaea. Recall that you will find cell wall in bacteria and you'll find them in archaea too. Archaea cell walls can be very diverse, but they don't have this large polymer called peptidoglycan. You will find peptidoglycan in the cell wall of both gram-positive and gram-negative bacteria. Archaea membranes typically contain either linkage instead of ester linkages. You would find ester linkages in the membranes of bacteria and eukarya. Some archaea have a lipid monolayer instead of a bilayer. Turns out that a monolayer, as well as some other structural differences of archaea membranes, can be useful for surviving in extreme environments. You will also find a unique cell wall in archaea. Recall that you will find cell walls in bacteria and you'll find them in archaea too. Archaea cell walls can be very diverse, but they don't have this large polymer called peptidoglycan. You will find peptidoglycan in the cell walls of both gram-positive and gram-negative bacteria. While archaea can also technically be gram-positive or gram-negative, most archaea staining gram-negative. Archaea don't have the peptidoglycan. Archaea can have pseudo-peptidoglycan though, which is a little confusing, but it has a different chemistry to it. Like all organisms, archaea have genetic material. Now, similar to bacteria, archaea DNA is generally arranged in a circle. Although, while bacteria tend to have one origin of replication, archaea tend to have several. When studying the genetic of archaea, it's interesting that now scientists find more similarities between the genetic of eukarya and archaea and than they do archaea and bacteria. So overall, these were some comparisons of archaea with bacteria. Keep in mind that most archaea can't be cultured in the lab. That's because there's a lot unknown about what kind of media they need to grow on, or nutrients or environmental controls in order to grow them in the lab. Although that's a common challenge for quite a few types of bacteria too. Before we go, I want to mention my very favorite Archaea genus. So, I have not been to a volcanic spring. Need to travel more, but Archaea members of the genus Sulfolubus have. They can handle temperatures that most organisms can't, like around 80 degrees Celsius hot. And that's not all. This genus can also handle very acidic conditions. Substances such as hydrogen sulfide, which is dangerous to most living organisms, does not deter them. There's interest in studying how this archaea could be used to treat industrial waste and even convert parts of that industrial waste into something that could be useful. That's all for Archaea. Thank you for listening.